Hi, I'm Steve Palumbi, and this is lecture 3 in the Science of the Extreme, Life of the Sea, where we're going to talk about the smallest organisms in the ocean, the microbes. The previous couple of lectures talked about the evolution of life on Earth, and then we sort of mentioned that the first couple of billion years of life on the planet, microbes had evolved and were the dominant life force in those particular times. Well, now we think about the life of the ocean as being mostly dominated by big things, the fish, the whales, the sharks. But in fact, microbes still are the most abundant, the most diverse creatures in the ocean, and they still play a huge role in how the oceans work as ecosystems. So this gives a kind of size scale of life in the ocean. We start here with zooplankton on the millimeter scale. These are copepods, for example, small crustaceans that graze the phytoplankton that live in the surface of the waters. But also in zooplankton are fish larvae and the larvae of other invertebrates. A little smaller than that are the phytoplankton. These are single-celled eukaryote and single-celled microbes that photosynthesize. They are in the realm of a couple of microns and then up to tens of microns. Then the bacteria, or the microbes themselves, are in the micron size. This includes the eubacteria, the archaea, photosynthetic bacteria, and bacteria that consume organic carbon, so-called heterotrophs. Below that bacterial size are the viruses, down in the submicron to hundreds of nanometer size. And then below that, there's size ranges that are not, strictly speaking, live things, but are made up of dissolved organic carbon. Dissolved organic carbon, the really tiny molecules are down in the nanometer size. They're truly dissolved, but sometimes they glob together to form colloids and larger accumulations of organic molecules, and that colloidal set of sizes is on the range of nanometers to tens of nanometers and above. Well, we didn't know very much about the microbes in the ocean, mostly because we couldn't see them all. Now, when microscopes became better and better and better, you could see microbes. Yes, we could know that there were microbes in the ocean, but it wasn't until the 1970s when stains were invented that stained the DNA of organisms and caused the DNA to fluoresce blue, that all of a sudden the huge kaleidoscope of different kinds of critters became apparent, even just a single milliliter of ocean water. This is a fluorescent microscope shot showing chains of microbes here that are all showing up with their fluorescent stained DNA. And by staining the DNA and counting the number of cells you see in a milliliter, scientists could finally get an accurate count of what the microbial life of the ocean really was. The microbes in the ocean tend to be in the range of about 1 million per milliliter. And if we begin to then take that number, which is a pretty big number in a very small volume of ocean, and expand that up to the size of the whole ocean, then we can get some rough idea of how many microbes there really are. And when we go through these calculations, the 321 million cubic miles of the ocean and how many milliliters there are in a cubic mile, go all the way down to the number of gallons, the number of liters, we get to an estimate of something like 10 to the 29th microbes in the world's ocean. That's an enormous number. To give you an idea how big that number really is, we can look at some other big numbers. For example, the number of stars in the known universe. Astronomers have taken the number of estimated galaxies, which is 10 trillion, the number of stars in a galaxy, multiplied those together, and even if this is an underestimate of what the number of stars in the known universe are, it comes out to be something on the order of 10 to the 24th. So 10 to the 29th microbes, vastly larger than the number of known stars in the universe. Well, that actual set of microbes in the sea means that they play an outsized role in the kinds of production that's there. This gives you an idea of a little bit of the ecosystem of the microbes in the ocean. It's not like they're floating around individually by themselves with no association with any other structures. This shows images of free bacteria, some phytoplankton, those are the photosynthetic single cells, protozoans, single-celled organisms that have a nucleus and are eukaryotes, and they're associating with one another and with the colloids, the colloidal dissolved organic carbon that I was talking about before, for example, in these kinds of globs. Marine microbes often accumulate on small bits of organic matter. They're some of the main reasons why that organic matter is eventually broken down, its contents released back into the open ocean, and the sort of ghosts of that organic matter and the marine microbes that are living on it are what's called often marine snow, because it falls very gently and slowly deeper and deeper into the ocean, eventually hitting the bottom of the sea. These small organisms are very abundant. They also play a big role in the metabolism of the ocean, or the whole way the ocean moves through, or the whole way that carbon and energy moves through the oceanic food chain. This is a graph showing the relative biomass of individuals down to about 10 to the minus 14th grams, up into the hundreds or millions of grams. And the larger the organisms, the bigger they are, obviously, but the lower their metabolic rate is per individual. Large organisms have a lower metabolic rate per gram of tissue than small organisms do. So you take the higher metabolic rate of small organisms and multiply that with a very large number of those small organisms. That together is the total combined metabolic capacity of that kind of organism. And the bacteria, the protozoans, the smaller ones, are up here in terms of the annual production of carbon in the ocean. The other critters we think of mostly as ocean life, invertebrates, fish, even marine mammals, bigger individuals, much lower on the scale of annual production in the ocean. We begin to sort of add that all up. The amount of organic matter in the ocean in these different categories, and a lot of it is in the microbes. So if we think about photosynthesis in the ocean, how much CO2 is actually fixed by photosynthesis into larger chain organic molecules, it's about 60 gigatons every year. That's about how much CO2 is fixed on land. So the photosynthetic production of the ocean and the land are about the same. A little bit of that, about 0.3 gigatons, drops to the ocean floor, often in this marine snow that I was talking about. A lot of it, 600 gigatons, is held as this dissolved organic molecule trove in the ocean water of the world. And that's a huge stockpile of organic carbon. It's about a quarter of the entire stockpile of fossil fuels in the planet, just the dissolved organic carbon floating around in the ocean. So just in terms of numbers, microbes play a big role. How are they actually involved in the life of the ocean, in particular marine food chains? And what's shown here is sort of a typical food chain. We tend to think about the bigger organisms, the fish, for example, that are consuming smaller fish, and then the grazers like the zooplankton. Shown here are copepods that are grazers mostly of phytoplankton in the ocean. But below that is this entire bacterial microbial mass that has a huge amount of metabolism going on. And then on this side are the viruses and other organisms that are consuming this bacterial population. So we have this part of the ocean food web, which we tend to pay more attention to, but then there's this other part of the ocean food chain. Both of them intersect in the bacteria. Depending upon how much of the carbon energy of the ocean goes into this part of the food chain, that changes how much is left to go up in this part of the food chain. So if we go to places like, for example, the Eastern Mediterranean Sea, there's a lot of productivity, but it doesn't have a lot of fish mass productivity, because a lot of the productivity of that particular oceanic mass is directed into the viral side of this food chain, with viruses consuming and killing the bacterial population, less of it able to go up into the fish. By contrast, if we go to a different place like the Gulf of Maine, this side of the bacterial food chain is suppressed, and a lot more of it goes up into the grazers and then the fish. So in different parts of the ocean, the same food chain is operating, but it tends to actually be one side or the other, depending upon the circumstances of that particular part of the ocean. Along the way, bacteria are producing a huge amount of biomass day by day by day. There's about 11 billion tons of bacteria in the sea. Suppose they're doubling about every 7 days. That is about 1 million tons per minute of organic production from microbes in the ocean. To give a little perspective on that, global fishing and aquaculture take about 100 million tons of food out of the ocean, call it 90 million tons, and without so much of the aquaculture counted. So that 90 million tons takes us an entire year to gather. It would take bacteria 90 minutes to generate the same amount of productivity as that 90 million tons in the annual fisheries catch budget. So far I've been talking pretty generally about microbes as a group. But I want to spend a little bit of time talking about individual types of microbes, in particular Prochlorococcus. So reason for that is that it has some unique features. It's one of the smallest microbes, less than a micron. It's also one of the most abundant types of microbes in the sea. Estimates are up to about 10 to the 27th cells of this one type of microbe in the world's oceans. And if you remember the estimate of 10 to the 29th, microbes in the whole ocean, that means about 1 in 100 microbes is this one type. They're photosynthetic, and because they're very common and they're very metabolically active, they produce about 50% of the oxygen in the ocean. That allows lots of the rest of the critters in the ocean to breathe, but it also gets into the atmosphere and eventually builds up the stockpile of oxygen that we use to breathe. And as people had looked at this one big, huge type of microbe, they began to realize that, in fact, even though it was called the same thing, that there were various strains of it that were adapted to different types, different parts of the ocean. In particular, shallow water parts of the ocean get a lot of light, but as you go deeper, the light dims, a lot less light deeper in the ocean, and the light is actually a very different spectrum the deeper you go. And if you start taking populations of Prochlorococcus from different depths of the ocean in particular, you can find very different types of organisms there. This just shows some of the different types there are, and it introduces a way of looking at the relationships among organisms we're going to use a lot. This is called a phylogenetic tree, or you could call it an evolutionary tree. And the way it basically works is that you have this set of lines that connect these entities here. Each of these represents one population or one strain of Prochlorococcus. HL means high light. LL means low light, and so this is a population of cells that were taken from a high light environment. What is done with these is that the population of those cells is strained out, the DNA is extracted, parts of the DNA are sequenced and compared to one another, and then the comparison of those DNA sequences are used to draw this tree. Things that are most similar to one another in that DNA sequence occur more closely together on this tree. So HL1, HL2, and HL6 are pretty closely related to one another in this part of the phylogenetic tree. And that means that they probably are evolutionarily much more related to one another. They diverged from one another not a very long time ago. By contrast, down here in this part of the tree are different types of the Prochlorococcus. In particular, the low light level Prochlorococcus are all down here. And what this tells us is that even though they look very similar, and even though they're not that different in terms of where they are, just a little bit of depth difference, that there's actually a pretty large evolutionary difference between the high light Prochlorococcus and the low light Prochlorococcus. This kind of tool, a phylogenetic tree based upon traits, often DNA sequences, we'll be using a lot in order to distinguish different organisms from one another and to get an idea of how they have evolved, and in this case, how they specialize on one type of environment versus the other type of environment. So now in this section of this lecture, we're going to basically switch gears a bit, and we're going to talk about a different kind of impact of microbes on the ocean environment, in particular using the deep water horizon oil spill as an example of the incredible and sometimes very surprising metabolic power of microbes in the world's ocean. So the Deepwater Horizon spill released hundreds of millions of gallons of oil into the Gulf of Mexico. It was a pretty deep oil well, about 1,500 meters deep, and it essentially spewed out oil over many months. The oil, some of it was just dispersed and blown downstream on current patterns. Others, like the bigger droplets, rose up to the surface where they formed scum and skims on the surface. Lots of efforts went into trying to gather that up, either by burning it off or using booms or ways of getting those droplets all together, dispersed by chemicals. And yet, even although those efforts, most of the oil that came out of the Deep Horizon oil well wasn't cleaned up by human activity. In fact, most of the oil that came out of this well was cleaned up by microbial activity that happened over the months that the whole oil spill was going on. So you know the microbes during the Deepwater Horizon oil spill changed in their metabolism, what it is they do. But how do we follow microbial communities over time, particularly in this sort of setting? Well, what we do in order to look at microbial communities is to take a sample from the water, filter out all the microbes, and then extract the DNA from those microbes. With that DNA, we can do a lot of different things. One relatively rapid way of getting an idea of the community is to amplify or to copy certain genes out of that mixture. In particular, the 16S ribosomal gene is universal among microbes. We can amplify it, then take that those amplification products of everything that was in a sample, put that on a DNA sequencer, and get out the sequences of the different kinds of microbes that were in that original sample. We can also take that same sample and extract not the DNA, but the RNA, the messenger RNA, which is the RNA that gets then transcribed into proteins. Take that, also put that on the sequencer and get an idea of not what the bacteria are, but what genes they're expressing, that is, what their metabolism is like. And by having these two kinds of information, the 16S gene, which tells us what kinds of bacteria they are, and then their metabolism, what we get from the transcriptomics, then we have an idea of what's there and what it is they're doing. So the 16S genes, how do we use those to actually get an idea what the bacteria really are, the microbes? Take that, get its sequence, and then compare that sequence in a phylogenetic tree to other sequences in the databases that exist. With the phylogenetic tree, as I mentioned a couple of minutes ago, allows you to look at groups of microbes, groups of sequences, they're called clades, and then look at the relationship among those groups, the relationship among those clades. So for example, here we see sequences from the international databases, GenBank in particular, and the sequences are formed into a phylogenetic tree. For example, this is a clade of the family Coeliaceae. This is a clade of bacteria that are mostly symbionts in mussels that live in deep sea methane seep habitats. And this is a clade of of microbes that are called methanotrophic. That is, they are able to take methane and use that as a food source. Now, suppose you had a 16S sequence from some sample in the ocean. You fit it into this phylogenetic tree, and depending upon what clade it fell in, you would know what kind of bacteria that was, and you get an idea of the kind of metabolism that that bacteria had in relationship to the other organisms around it in that clade. Well, when that was done with the samples from the deep water horizon oil spill, then we can look at changes that have occurred over time. In this particular graph, the oil spill starts here, and in the plume, the water plume from the around the spill, we see the growth of one group of microbes called the Oceanospirales here. Became very common and died off. Colwellia started out small and got large. The methylotrophs started very uncommon and then got very common. And then there's flavobacteria and rhodobacteria that became much more common over time. So these diagrams are a way of taking the data from the samples and then the 16S phylogenomics, and then looking at the composition of the community over time in the plume. You can also do that in sediment samples. This is deep sea, and these are coastal sediment samples around that part of the Gulf of Mexico. So that gives us a way of thinking about how the bacterial population is adjusting to this oil spill as food, and the kinds of bacteria, the kinds of microbes that grow up to be able to consume it. Well, the other thing that you can do is to look at the metabolism of those microbes over space and over time by looking at the genes that they're expressing. This particular analysis, they're looking at the genes that transport large organic molecules from the outside to the inside of the cell. That's what's necessary in order for the cell to actually start consuming them and converting them into energy, those transporters. This diagram shows the abundance of transcripts, or the abundance of genes in the messenger RNA that transport a bunch of different kinds of organic molecules, amino acids here, for example, taurine, urea, phosphate there. The size of the circle tells you how dominant that particular transporter is in this sample of microbes. And then from the top to the bottom here, we're looking at the euphotic zone, pretty shallow areas, all the way down to 4,000 meters deep. And when you do that, you can see big changes in the kinds of metabolism that are being done by these microbes over depth and over the different kinds of molecules that they are able to transport inside themselves to consume. So the combination of this tells you a little bit about the metabolism that's going on at different depths and in relationship to different food supplies. And then the 16S data tell us a little bit about the names and the taxonomy and the clades that those microbes are in. And those are some of the tools that people are using in order to try to understand not just the number of microbes and their overall role in the ocean, but chop it down a little bit more finely, the kinds of microbes they are and what it is they're doing. Another aspect of bacterial genomics that happens in the ocean and in soil and in lots of other cases is entire genome sequencing. Not just a small 16S region, but the entire microbial genomes can now be sequenced and compared to one another to give you even greater resolution of the phylogenetic tree. We use a huge amount of fertilizer in farms to grow better crops, but a lot of that fertilizer essentially leaks out of the fields into streams and rivers, down into estuaries and bays, where it still acts as a fertilizer, causing huge blooms and microbes in coastal waterways. Those blooms and microbes sometimes get to be so dense that they use up all of the available oxygen in that water. That kills everything. That allows other microbes to come in that use anaerobic metabolism to consume the dead organisms that were there. And more and more of the bacteria then consume the oxygen. I'm going to start over again. Dead zones in coastal Dead zones in coastal oceans are a recurring and annual problem that we can see all over the world, and it comes from essentially our use of fertilizers in farming. We use a lot of fertilizers in farming, makes crops grow a lot faster and bigger, but a lot of that fertilizer leaks out of the fields into streams, into rivers, down into estuaries and bays, where it still acts as a fertilizer, and it causes huge blooms in microbes in coastal oceans. Those microbes become so abundant that they use up the oxygen in that water, killing everything that's in it, and that's why they're called dead zones. One of the best examples of the role of dead zones in changing the ecology of an entire ocean basin actually comes from the Black Sea. Here's an image of the surface of the Black Sea, and you can see in it this set of plumes. These are microbial blooms that have resulted from fertilizer washing in. That microbial blooms eats up the oxygen, causes the dead zones. And those were so recurrent and so intense in the Black Sea that the entire ecology changed. So to give an example of what's normal, the Black Sea, like big ocean basins, a wide variety of different fish and fisheries for anchovies and tunas, etc. In the surface areas, lots of oxygen, moderate amounts of oxygen in the middle zones, and plenty of oxygen in a diverse set of bottom-dwelling organisms down in the deeper zones. But when, in the 1970s, the huge influxes of fertilizer came in, then that caused a huge drop in oxygen. The zooplankton and the phytoplankton in the surface changed. The water in the middle of the Black Sea, the middle layers here, became very poorly oxygenated. Up above, there was some oxygen still able to come in from the atmosphere. Those organisms began to die because of hypoxia, fall to the bottom where they're consumed by even more microbes, making the oxygen levels even lower at the bottom of the Black Sea. As that whole picture progressed in the 1980s, the fish and fisheries populations more or less died off, and that resulted in a bloom of jellyfish as a way of something coming in and consuming the phytoplankton or the zooplankton that were living in the surface waters. The ecology then of the Black Sea shifted from a normal fish-dominated food chain to one dominated by hundreds of millions of jellyfish that made up most of the biomass of the surface area of this body of water. All those changes happened because of microbes. All those changes happened because the populations bloomed. All those changes happened when the ecology was shifted by the chemical infusion of fertilizers into those areas. And it just shows the power of microbes in changing the way the ocean actually works as an ecosystem.